Today, we delve into a series of fascinating and critical studies published in Animal Genetics and Genes, focusing on a group of devastating inherited neurological conditions in dogs, specifically the neuronal ceroid lipofuscinoses, or NCLs, and related neurodegenerative disorders. These conditions are characterized by progressive deterioration of cognitive and motor function, often leading to severe quality of life issues and premature death in affected animals. Our first paper, by Bellamy and colleagues, describes a case of neuronal ceroid lipofuscinosis in a Chipendo's dog, highlighting a novel genetic variant and its implications. The broader field of lysosomal storage diseases, to which NCLs belong, represents a heterogeneous group of disorders where the body's cells fail to properly break down and recycle waste products, leading to their accumulation, particularly in neurons. This accumulation results in the characteristic neurodegenerative signs that we see in affected dogs, which often mirror those observed in human patients, making these canine models incredibly valuable for understanding disease mechanisms and exploring potential therapies. The research we're discussing today, conducted by various teams, aims to pinpoint the specific genetic mutations responsible for these debilitating conditions in different dog breeds, offering crucial insights into their pathogenesis and paving the way for improved diagnostic tools and breeding strategies to prevent future cases. The clinical presentation of these diseases can be varied, but typically involves a constellation of neurological deficits, often starting in early life and progressing relentlessly. Common signs include visual impairment, seizures, changes in behavior and cognitive abilities, and difficulties with movement and coordination. Pathologically, these disorders are often confirmed by the presence of abnormal intracellular accumulations of autofluorescent lipopigments and a significant loss of neurons within the central nervous system. These pathological hallmarks are central to diagnosing and understanding the specific forms of these neurodegenerative conditions.
Building on that, the Bellamy team's findings in the Chipendos provide a clear example of how these genetic discoveries are made and their profound impact. They investigated a 19-month-old Chipendo's dog that presented with clinical signs highly suggestive of a lysosomal storage disease, which was subsequently confirmed by pathological examinations consistent with neuronal ceroid lipofuscinosis. Specifically, the post-mortem examinations revealed typical histopathological findings, such as the accumulation of autofluorescent cytoplasmic inclusion material, which strongly supported their initial diagnosis. To uncover the genetic basis, whole genome sequencing was performed on the affected dog and both of its parents. This in-depth genetic analysis led to the identification of a missense variant in the CLN6 gene. Thank you for listening to the Veterinary Medicine Digest. Remember to like, share, and subscribe as proceeds from this channel go directly to providing vital veterinary care for animals in need.